first things first, thank you so much for all of you coming and being part of our audience. Part of being in an audience is like a regular classroom. You do not have your phones out. You are not talking while other people are talking. If this is not something you think you can handle, the door is there. I'm certainly not going to force you to stay, because my class has actually put quite a bit of effort and time into trying to prepare for this. So we're going to ask for your cooperation. Understood? Okay, good. All right, so we do this presentation every year. Oh gosh, this is like 18 or 19th annual College Bio Current Event Symposium. So we've been doing it for quite a while. The reason we started it is because so much of what we do in the college bio class is old sort of foundation biology. Very, very, like, what did they learn in the 17 and the 1800s? How does that, transcribe to the current things that we're doing today? But we don't get to do cutting edge. We don't get to do stuff that happened in the last year and a half. Like, that's just not how the course is presented. So what I like is that we do a lot of foundation, and then you get to do this research assignment that allows you to sort of build off of what we've been learning over the last, 3/4 of a year. Students are allowed to pick any topic that they like. So today, it's a little dreary. They all picked topics on cancer, but cancer is a big deal. It is always in the news, so there was a lot of information out there. There's a lot of new technology and a lot of new research helping to try to diagnose and cure, or at least to better understand how cancer exists within the body. I do want to thank you all for coming. I'll give you a little bit of an idea of what we're looking for. The students have made a presentation. They've made slideshows. They have prepared some things to say. They have a couple comments. Their presentations should fall within 4 to 6 minutes. You'll see me in the back. I'll be timing them. Like, there is a goal. If you are over or under, you lose points. So there's a sweet spot that they're really trying to aim for. That's, that 4.5, 5 minutes. We encourage you to be listening. And if you want to ask a question at the end, we will open up the floor, the audience. Presenters, the best way to do questions, because we are being recorded, and I know that they like to put this on TV, is when someone asks you a question, to repeat their question and then give the answer, because sometimes when the audience doesn't have the microphones, it doesn't translate well. So when you hear a question, repeat it and then provide your answer. I think we're good to go. We're really excited. Our first presenter is Sarai Cruz.
Why are some animals less susceptible to cancer than humans? As we know, cancer is the second most leading cause of death in America today. Cancer is the result of mutations in DNA which cause cells to grow rapidly and uncontrolled. The p53 gene, a tumor suppressor gene that makes protein found in our cells and humans, is a pivotal gene in order to control both cell division and cell death. This gene searches for cells with miscopied DNA. There is another gene known as the LIF6 gene, or leukemia inhibitory factor, that is used to obliterate damaged cells. It was said to have just disappeared in elephant DNA millions of years ago, but evolutionary analyses indicate that LIF6 is a refunctionalized pseudogene. So why is it that animals with higher cell count and longer lifespan are less likely to develop cancer than humans? This is because bigger marine and terrestrial animals with more cells have more suppressive genes stored in their cells. Large body sizes naturally activate a larger quantity of suppressive genes, where our human bodies don't use them as efficiently. This phenomenon is referred to by scientists as Peto's paradox. How does it work? When cancer affects elephant cells, they use their suppressor genes just as humans would. The difference is, humans only possess one copy of the p53 gene, but elephants have two. Being that LIF6 is only present in elephant DNA, Humans have no gene to eliminate cells affected by cancer. LIF6 latches on to the mitochondria and chews at them, forcing the contents out, killing the cells. So the P53 gene occurs 20 times in elephant genome. The process. All mammals have one LIF gene and one promoter that helps activation. Elephants, however, also have dormant copies of the LIF gene, also known as pseudogenes or the LIF6. Elephants actually have 8 copies of the LIF gene, but only LIF6 is proven to be functional. When the dormant cells reactivate, proteins from the P53 gene create protection, meaning it had become functionless after being incorrectly communicated. We don't know how the gene developed a new on switch, but ultimately it brings the gene back to life. How can this help us? With this information, we can explore new possibilities for new methods of diagnosis and cancer detection in humans. Currently, at the Vienna Zoo, blood samples are being taken from an African elephant in order to observe how P53 proteins react with damaged cells, and then they're able to note the similarities and differences with results from human cells. And currently, scientists are working on finding ways to develop medications that mimic the behaviors of LIF6. We have yet to see any success. Also, I have a little fun fact. So, taller humans are at higher risk for cancer because they have more cells but still only have one master suppressor gene. My little fun fact.
For some people.
So really interesting when you think about it. You've got a gene that does, we talked about in college bio, we talked about the suppressor genes during the cell cycle, and we talked about it regulating division. With the elephant, are they looking to, in your presentation, maybe I just missed it, are they looking to add the elephant suppressor gene to the genome, or just to make copies of the ones that are human?
Well, what I looked at was they're trying to mimic like the gene that the elephants have, the LIF6 gene, they're trying to replicate that with medication. So to treat humans with cancer, if they can replicate that, and then it'll treat it better and kill off those cells.
Does anybody else have any questions for Sarai? Yes.
So I see on your cover page there you have whales. Has this been studied in whales also?
Yeah, but there wasn't as much information on it. In the article that I was researching in, it didn't give much detail on the copies of the P53 gene as it did with the elephants. I think they've done more studies in that area.
Nice job, Sarai.
When should dogs be screened for cancer? Background information. Cancer is the leading cause of death.
Very cool.
Cancer is the leading cause of death in adult-aged dogs. Cancer screening for dogs rely on really invasive procedures, like surgical procedures, to remove the tissue, and that's not really as effective as something like a liquid biopsy would be, because it is less invasive for the animal itself. Liquid biopsies have increased the ability to diagnose cancer at earlier stages in dogs. Now I'm sure you're wondering, what is a liquid biopsy? A liquid biopsy is a form of blood testing that can help detect and diagnose cancer in dogs and in humans. A A sample of blood is taken from the dogs, and researchers look for fragments of DNA that could detect a cancerous mass. With the help of non-invasive liquid biopsies, researchers were able to shift their focus from how to test dogs for cancer to when should we test dogs for cancer. Of 3,452 dogs, 2,537 were purebred, and 915 dogs were mixed breeds. And this is crucial to have a diverse amount of both in a proper scientific study. 55% of the dogs were male and 45 were female. The dogs had a median weight of 30.6 kilograms and a median age of 8.5 years. Common cancer types in this study included lymphoma, osteosarcoma, mast cell tumors, and soft tissue sarcoma. In this study, weights were only available for 2,551 dogs. The weights ranged from 2.5 to 80 kilograms, or to 98 kilograms, with a median weight of 30.6 kilograms. For specific breeds such as bulldogs, boxers, Boston Terriers, and French bulldogs, the median age for cancer diagnosis was less than 2 years younger than the projected median age that was predicted by the weight-based model. Researchers noticed that there was not a large difference between the age for cancer diagnosis between non-neutered males and females. Dogs who had been neutered showed later ages of cancer diagnosis than their intact counterparts. There was no interaction observed between neuter status and weight in the dogs. How does this research benefit the average person? This research benefits anyone who has a dog, regardless of their occupation. It is important for dogs to receive proper medical care, and with this research, people can know when to start getting their dogs tested for cancer based off of specific genetic information. The more that is understood about this topic, there is a greater cause of dogs being able to receive the best medical care they can receive. This study opens up a lot of new conversation and ideas, and these are some of the questions that I came up with. If a dog's breed and weight can determine when they should get screened for cancer, could this research be applied to other animals? And if so, could this be applied to non-domesticated animals other than cats, like wild animals? And could there be other genetic components that we should be looking out and testing for with cancer that could help this research evolve? Researching for when dogs get screened for cancer is crucial for dogs to be able to live their best life and be as healthy as they can be. This research promotes the conversation on when dogs should begin to be screened for cancer based off of, their weight, sex, age, gender. The idea behind this research is incredibly helpful for their dogs and their owners. And this is my works cited page.
Do vets know about this? Like, is this something, like if I have a dog, he's a little crazy, but if I felt like he fell into the appropriate parameters, like, is this something that's common? Is this like this information is out there where like the average person can go to their vet and use this as an impetus to have their dog tested?
Well, this research is fairly new. It did come from this year, 2023, but I believe you should be able to bring it up to your vet and be like, is this something that I can use to help my dog?
Did you see anything on what breeds are more likely to achieve cancer, like pure breeds versus mixed dogs?
Yes, I did. So purebreds are more likely to develop cancer as to something of a mixed breed dog. Yeah, because of the genes, like when you're breeding a purebred dog.
Okay, my presentation is about how ants could potentially become a new method of cancer detection by just using their antenna. How does this work? Although ants lack noses, they have an abundant amount of olfactory receptors that are located on their antenna, which allow them to basically smell. Ants have about 4 times as many odor receptors as other insects, which lets them get through their daily lives, like sniffing out food or like mates. And scientists have determined that this could be used to detect cancer cells in humans. Tumor cells release a chemical called volatile organic compounds, which makes them able to be detected in things like sweat or urine. And other animals that have an incredible sense of smell, like dogs, have previously been trained to detect cancer cells in things like body odor or saliva, but scientists want to extend their research to see if ants could do the same thing. The experiment. To begin the experiment, the scientists first started with transplanting a human sample of breast cancer into mice through a process called xenografting. After letting the sample grow inside of the mice, urine samples from both infected mice and unaffected mice were collected and placed inside of a petri dish with the ants. And to train the ants, the scientists used sugar water. They placed the sugar water in front of the cancerous urine samples to get the ants to associate the smell of the cancer urine with the sugar water. The results. After just 10 minutes, which was about 3 rounds of training for the ants, they were able to successfully associate the cancer urine with the sugar water. Once the scientists removed the sugar water from in front of the urine samples, they stayed around the cancer samples for around 20% longer than they did the other sample. And the ants were able to successfully do the experiment up to 9 more times without receiving a reward, so they were able to accurately do it. How can this benefit people? In the United States, cancer causes every 1 in 6 deaths, which makes it the second leading cause of death after heart disease. In 2021, cancer alone killed about 10 million people, and usually the key to a more successful treatment is detecting the cancer early on. Currently, many early detection methods are unavailable to people, especially those without health insurance, because of the cost. However, the cost of training and maintaining these ants is significantly less than other cancer detection methods, which could potentially make this both reliable and cheap for the patient. It is also more efficient to train the ants compared to the dogs, because the dogs can take up to 6 months to train, while the ants only took 10 to 30 minutes. But although the scientists are very hopeful about the direction of this discovery, a lot more research has to be done, because this was just discovered in January, so it is very new. So a lot more research has to be done. What new questions does this experiment bring? While the experiment worked with urine samples from mice, it is unclear if it will even work with human urine samples. Experiments using human urine samples that are infected with different types of cancer and not just breast cancer will be done to test the ants' abilities. Scientists also will experiment with the size of the tumors used, because the size of the tumor transplanted into the mice was abnormally large compared to their bodies. And the scientists want to see how small of a tumor can the ants detect before they're no longer able to differentiate the unaffected urine with the infected urine. Finally, normal human factors will need to be considered, like age, sex, weight, and diet, because it's different for everyone, and this can affect the results. And more research that includes these factors needs to be done to test if this is a legitimate method or not. Thank you.
So do you think that they have started to compare you mentioned dogs, which I've heard dogs are also good at sensing, cancer, among a variety of things, but they can sense cancer. It took 6 months to train a dog, but then a dog will, live for years and years.
Yeah.
I'm assuming with ants, they had 9 times, and then can you retrain them? Do you think? Did they say that they could do that?
They did not say anything about that, but there's so many ants in the world that you could just I don't know, go in your backyard or something, ants, and train them to, But I'm not sure if you can retrain them, but they were able to redo the experiment many times accurately, so it's
Right.
possible.
So I know you mentioned they only did one tumor size,
Mhm.
but did they do it with one type of cancer, or were there multiple cancers they tried?
From what I read, they only did breast cancer, but they want to do more experiments with different types of cancer to see if it can work for everything, because there is a lot of cancers that don't have like a detection method, you know what I mean? Like you can detect like breast cancer early on or like some other forms of cancer, but there are a lot of cancers that you cannot detect early on, so we want to see if maybe this could solve that problem.
You know, speaking of hard cancers to detect, the legacy of pancreatic cancer. The initial question, what causes pancreatic cancer to be so resistant to conventional cancer treatment plans? The key percentage, 10 to 11%, is the 5-year survival rate of those diagnosed with pancreatic cancer in the United States today. Please keep that in mind. Define treatments. Pancreatic cancer stem cells. Pancreatic cancer stem cells are highly aggressive cancer cells that resist conventional therapies and encourage tumor relapses. They rely on epigenetic regulation, which is the changing of characteristics of a cell due to altered gene expression, in order to protect themselves and promote tumor survival and growth throughout conventional treatment tactics. Continued research is to find the underlying mechanisms that cancerous stem cells are using. Identifying how the resistance is truly occurring helps to then figure out how to work around and against it. SWI-SNF is a family of proteins. These proteins regulate chromatin, which is a mixture of DNA and proteins that form chromosomes required for stem cells to function properly. These subunits are often known to be tumor suppressants. This is where SMAR-CD3 comes into play. As a part of the SWI-SNF family of proteins, it was found to be amplified in pancreatic cancer cells. Throughout modelings of pancreatic cancer, this protein was deleted. And as a direct result, tumor growth was reduced and remission was improved when paired with chemotherapy, a quite conventional treatment option. The reasoning. SMAR-CD3 has been found to control lipid and fatty acid metabolism, which is found to be directly associated with the resistance. This reason being is that it provides a metabolic landscape that allows pancreatic cancer cells to avoid anti-cancer treatments and grow rapidly. This roots from lipids being a source of energy, feeding the cancer cells, driving the rapid growth and spread that normally is associated with pancreatic cancer tumors. Benefits for citizens. This research can help the average citizen understand what they or loved one is going through internally. The potential therapy this research can facilitate increases the hope and possibility of remission. It has the potential to lead to save lives, and it brings awareness to the lives lost and ones that can potentially be saved. Conclusions. This all flags SMAR-CD3 as a new target for therapies. All this research is lending way to target the third deadliest cancer in the United States, saving lives and increasing the astonishingly low survival rate of those diagnosed with pancreatic cancer. Now, the question remaining for researchers is how to attack SMAR-CD3 directly inside of the human body, now that we know it's a major cause of pancreatic cancer resistance and in turn fatalities.
So you said that the genes are about, the genes you spoke of are about dealing with metabolism of lipids and carbohydrates?
Lipids and fatty acids.
Lipids and fatty acids, okay. Is there a way, is it your understanding, or was it part of the research that if they can modify that gene in some way? Like we've got new technologies like CRISPR and other methods that can go in and alter small segments of genes. Have they isolated where that gene is within the genome so that they can specifically attack it? Or is that what we're still working towards?
From the sources, that is what we're still working towards.
Okay.
But I'm sure that with all the technology, it would be a possibility.
So with the pancreas being like a relatively small organ, any of your research say like why it's so deadly when cancer pops up there?
It's because it's located so remotely inside of the body that it's harder for the symptoms to be seen earlier on.
Did they find this gene in anything other than, I know you read about the pancreas, but did they suggest, we would look for a similar gene in some of these, the other organs that are more susceptible to cancer?
Yes, actually.
Thank you so much for attending. This is this the 19th annual College Bio Symposium Day 2. I really want to extend my congratulations to the folks who went yesterday. You did a fantastic job. They were really great. Got a lot of really positive feedback from the people who were here who watched it. So they all told me, and I said, well, I think you should just tell them. It was really well presented. A couple things, we did up the volume on the mic, but you want to make sure that you speak into the mic so that we can hear everything you have to say, and make sure, you guys did not do this, you have to repeat the questions so that the camera can pick up on what the question was as you give an answer. All right? As far as your audience, thank you audience for coming. We do appreciate that you are a cooperative audience, so we're hoping that you will just pay attention. If you're not interested in being here, you just don't need to sit here. You can sit at the other part of the library, but we don't really want you to be a distraction on phones and talking and things. Our class has put a lot of effort and time into preparing these presentations. So we want to give them, I'm glad that we have audience here. It's good to present in front of people that you may or may not know. And we appreciate that you are here, but I certainly don't want you to be a distraction if you're not comfortable being here. So here's the point for doing the symposiums. For those of you who have not been here, we do this every year. College Bio is a dual credit course through Ulster, so you take 2 semesters, you can get up to 8 credits. This is the second half, so everybody who's in the class has already completed College Bio 1, they're in College Bio 2. College Bio, from Ulster, they asked us to do a research assignment of some sort. First semester, I have everybody write a research paper. They thought in the second semester, nobody wants to write another research paper, so we would do a different form of research where we could find something we're interested in and then do a presentation on that and share it with our colleagues, classmates, and staff. So each of the topics that our students present today are self-selected. There's no, the only parameter I give them is it should be related to biology. And they can kind of take it in any direction they like. I do insist that it is something that is recent. A lot of college bio, a lot of the information that we do is that foundational, sort of old school, everybody knows this biology. It's sort of the basis off of which we study. But we spend so much time doing all those old basics, we don't get to talk about some of the new cutting edge stuff. So that's what this presentation really allows us to do, is to give really interesting insight into how what we're learning in class is still being used today, and what we're learning, and really for most of you, what you learned in living environment can be used to understand some of this newer research. So without further ado, we have a wide variety of topics for you this afternoon. We're going to start with Joe Torres, and he's going to talk to us about biofuels.
You hear me all right? So today I will be talking about biofuels, Earth's ally. First of all, what is a biofuel and where is it obtained? Biofuels can easily replace the fuels we use today to power engines and jets. It can be obtained from algae, corn, soy, and even excrements from herbivores. Now, where is it harvested? Generally, there are two categories we take biofuels from, both microalgae and macroalgae. Microalgae are the best for biofuels. They are easily refined into a fine oil, and overall, the components are much easily modifiable. Making it usable. Making it usable is a long process. First of all, we need to modify the components inside the algae that we modify. Generally, they remove all the non-fatty acid components and enforce many fatty components to make it into a nice thick oil, which can be used in biodiesel, jet fuel, and even 100 low lead, which you would find in small airplanes. Applications. So as I said before, it could be used in jets, small aircrafts, and cars. And now we are even introducing biodiesel, which allows us to use it on big trucks, tractor trailers, and overall it is a much more affordable option for these companies. Now for the results of biofuels. Now, biofuels are expected to cut carbon emissions by 50% alone for aviation. And some of the estimates even go up to 86%. But by implementing biofuels, we hope that we could cut the overall emissions in America by half. Now, aviation is by far the leading cause of carbon emissions in America. They make up to 60% of it. And when we implement this into even small aircrafts, it'll cut down in the civic area of aviation by 30%. And then when you put it into aircraft industries, it'll make travel much more affordable, and it will extend our travel by far into international flights, or even flights that could be into more remote areas because it allows more endurance for the airplane. So are there any questions about biofuels? Shahid?
So, fuel, as we all know, is very high in demand right now, correct?
Correct.
Would switching to biofuel be able to match that demand? Because we are harvesting oil from all over the earth. Would we be able to, fuel the same demand with, biofuel?
Okay, so to summarize your question, you're asking, will it be able to meet the demands that fuel currently holds?
Yes.
Okay. Yes, so because it is so abundant, like such as algae, wheat, corn, we could use it a lot. But being now since the 2020 pandemic, it is much harder for us to implement biofuels right now. This is because agriculture is a much sensitive topic, and we don't want to take food away from the locals. And so right now, it is a slow process, but it is a process being implemented slowly in industries. Ryan?
We talked a lot about the positive risks, the positive benefits of using biofuels. Are there any negative risks that we should be aware of?
Okay, so any negative risk of biofuels? Mm-hmm. Okay, so as touching on like what I said before, in terms of agriculture, that is what we are worried about, because we do not want to take food away from, say, like smaller villages, especially in more remote areas. And this industry, such as deforestation, we don't want to harm the habitats of the earth. So that would be the cons of it. Ms. Kamicker?
Are biofuels less likely to produce carbon dioxide emissions than what we would with normal, traditional fossil fuels? Are we still getting some of those CO2 emissions? Is it significantly better for the environment? I know it's an alternative energy, but is it better in any way?
Okay, so you're asking if it'll reduce carbon dioxide emissions. Okay. Yes, by far, significantly. And that's why the government wants to implement this slowly, because like I touched on before, it could cut emissions in half. And it will be a lot safer option, and they actually found that it made vehicles run better. So not only will our mileage be better, we will be helping the environment because there will be so much less pollution in the environment. Any other questions? Mr. Barton?
What's the difference in the cost between like unleaded fuel and biofuels, or like between, what's the comparison? Is it less, does it cost less to produce or more?
Okay, so the cost versus modern fuels to biofuels. Okay, so overall, biofuels right now would be a cheaper alternative. The reason being is because we can import it from pretty much anywhere. While like modern fuels now, we have certain oil refineries that import our fuels. So biofuels would be cheaper because it's just so easily accessible, and most countries would have this in abundance. Any other questions?
That's life.
Great job, thank you.
Okay, so before we get started, I'm Justin, for those of you who don't know, and I did my research project on the invasive aquatic species of water chestnuts. So I'll pass a few around. Just be careful, the edges are very, very sharp, so. So just like I said before, they are very sharp, so just be careful. So actually this picture here on this slide, I took it actually yesterday. It's Silver Lake in Woodridge, New York, and this lake is heavily infested with the water chestnuts. So many of you are probably wondering, what are they? Now, when I asked some of my classmates, do you know what water chestnuts are? They told me, oh, the ones from the store. But there's a very, very big difference in the ones you buy from the store and the ones you find growing throughout waterways. So they're called the European water chestnuts, and they're native to parts of Europe, Asia, and Africa. They were first introduced in the Northeast in the late 1800s because gardeners hoped that they could harvest them and then sell them to gain profit. In the more recent years, water chestnuts have become very invasive, as they're very hard to maintain as well as very expensive to control. And like I was just saying, their extensive growth patterns and their persistent seeds as they're being passed around make this plant very effective in invading habitats. So this here is the seed that's being passed around. This here is like the plant that these seeds grow off of. It's just pretty much a leaf, and I believe that they can grow from 10 to 16 feet and embed themselves in the bottom of a lake or a stream. So this here is an example of a whole ecosystem being invaded by them. As you can see, there's no sunlight going through the water because of how the leaves have grown. And now this here is an example of, this here is an example of the water chestnuts that are being passed around that I collected yesterday, of just how invasive they are on like a shoreline. So this can impact like any day activities such as fishing or maybe even swimming. So some of the issues they create can include that they grow in very large colonies. Typically, you don't see small ones unless they're just starting. So the large colonies, they like to compete against the native organisms, and so they like to compete against the native organisms for nutrients in space, which leads the ones that are already there to struggle to survive. And the water chestnuts can also lower the amount of dissolved oxygen in the water and negatively affect the current plant and fish species that are inhabiting this waterway. Because as most of you know, am I in the wrong slide? Oh. So as most of you know, fish and the plants do need dissolved oxygen in order to carry out basic survival functions. And then I was just saying before in the last slide how the water chestnuts were all along the shore. They can clog waterways, making fishing, boating, and swimming nearly impossible for onlookers. So overall, the water chestnuts do create social issues as well as environmental issues. So local impacts. I chose this issue because it's something that has impacted me, and it has actually a lot of local history around the area. So in Ulster County, there's a few different spots where they do impact, as well as the neighboring Sullivan County. So here I listed a few places, which include the Hudson River, Silver Lake and Woodridge, New York, where I just mentioned, Cape Pond, which is going up Ulster Heights Road in Ellenville, and Fort Binneywater, which is located in Rosendale, New York, heading out towards Kingston. And the picture I have there is once again Silver Lake, and it's kind of zoomed out compared to the previous picture that I showed of how that just impacted the shoreline. So this map here is of New York State, and all the green dots that you see are all the places where the DEC has reported them to the New York Invasive Species Information. And now I zoomed in on it over here, and I'm going to point it out. So Ellenville's right there. Right there, that one would be Cape Pond, and here is Silver Lake, and I believe up here is where Fort Binneywater is. But as you can see through this map here, this line here would be the Hudson River. Typically, these water chestnuts came from the Hudson River, and due to boaters not cleaning their boat properly, these water chestnuts have made their way to other areas. So, methods of controlment. So the large infestations usually require the use of mechanical harvesters or the application of aquatic herbicides. And as you see in that picture right there, that is, for example, one of the harvesters that they use. They come in all different sizes, but basically it's a conveyor belt, and it kind of chops up the leaves that are on the surface and a little bit down in the water. So, and then they do dispose of these offshore to prevent them from regrowing in the water, because just like any other typical plant, it does have a growing season where they will grow and then they will die off. And scientists do suggest, like if there's a small infestation, to hand pick it, but I'm sure most of you don't really want to do that. And it does take a lot of work, and it has just the same amount of chance of growing back. Now I just want to go back to how the aquatic herbicides, so they have experimented with 2,4-D, which is an herbicide that controls broadleaf weeds. And it's been used since the 1940s, but over recent studies, scientists have found that it's really not worth it, and it can impact other aspects of the environment. So this led scientists to turn to the use of biocontrol agents, which are organisms such as insects, or they might experiment with a plant disease that is used to control this and try to eliminate it from the population. So these are just some of the sources I got. My information from the DEC was very helpful because this is a statewide issue, and it's also an issue across the Northeast, as it's very hard to control, very expensive to control. Do we have any questions? Mrs. Goodmaker.
So, I see you said they chop up the plants. Do they have anything specific for harvesting seeds? Because my assumption is we've got that really sort of prickly protective coat on it, and it probably makes them float, and it probably keeps other things from eating them. Do we try to get rid of those as well, or are they just hoping that getting rid of the plant will eliminate the whole problem?
So your question was, do the harvesters also chop up the seeds? And to answer that, I'm not totally positive about this, but I have seen from experience, I do believe that it also works on chopping up the seeds as well as the leaves, because the seeds also is what help germinate the plant to reproduce. So I would assume that their best bet would be to get rid of the whole thing and disperse of it off the waterway to prevent it from regrowing. Next question, Mr. Duchamp.
So I know the past few years they've used these harvesters on the Hudson River, but they've had to go back each year and do it. Do you know if it's because the plants are growing back from their roots, or is it new seeds coming in?
So your question was the Hudson River, as I said before, with these harvesters. But how come they're harvesting these, but they're having to go back the next year and do the exact same thing? So to answer your question, it would be, you can't really get all of them with the harvester, and the ones that are left behind, when they do reproduce, they produce in large quantities. Next question. Ryan?
You said parts of Europe, Africa, and Asia, which is around 60% of the world's landmass. Where specifically do you mean when you say parts of?
So you said, going back to the general facts, how they're from parts of Europe, Africa, and Asia. I'm not sure entirely where they're from. They did come over to this country, like I said, in the late 1800s, so there wasn't very much information about that.
It would be somewhere in the northeastern climate, so would it be Mediterranean or be similar to something like
So to finish answering your question, it could be anywhere there. I'm really not entirely sure, but for this project, I really focused on covering how it locally impacted us.
That's fine.
Next question. Heather. Oh, never mind. I think that's it. Thank you, everyone. I'll come around and
Okay, my research project is on bear hibernation and osteoporosis. What is osteoporosis? Osteoporosis causes bones to become weak and brittle. When you have osteoporosis, mild stresses such as bending over or coughing can cause a fracture. Bone is living tissue that is constantly being broken down and replaced. Osteoporosis occurs in the creation of new bones and keep up with the loss of old bone. In humans, prolonged periods of inactivity as a result of bed rest or injury can cause rapid bone loss. Blackberries and hibernation. Bears experience unique aspects of hibernation that other hibernators do not. They're able to make sure their body temperatures do not drop as low as that of other hibernators, allowing them to react more quickly to danger. They also have cholesterol levels nearly 4 times higher than humans, but do not suffer from the hardening of the arteries or gallstones, which are both side effects of cholesterol levels. There is much to be learned from bears' hibernation habits, these just being a few unique aspects of the mechanism. How do they relate? Bears experience humans when subjected to long periods of bed rest fall to the risk of developing osteoporosis. However, black bears during their 4 to 6 months of hibernation are able to prevent this bone loss through multiple factors, another unique aspect of black bear hibernation. Anyone who has had a limb in a cast for a while knows how weak a human muscle becomes when it is not used for an extended period. But somehow, bears manage to spend months in hibernation and come out about as strong as when they went in. Black bears are physically inactive for up to 6 months per year, but unlike most mammals, bears do not experience negative effects of this disease period on bone structure and strength. So why don't black bears develop osteoporosis? Their mechanisms for bone loss prevention have been scarcely elucidated upon. However, we know that during hibernation, black bears are known to decrease their metabolic rates by about 20 to 75% to save energy, relying solely on the fast growth of their energy source. This new study shows that when bears reduce their metabolism, genes involved in bone restoration becomes less active. In comparison with other hibernating mammals, bears have a large body size, near to normal body temperature during hibernation, and a drastic pre-hibernation fat storage. Such peculiar differences may bring about bone preservation mechanisms that other hibernating mammals do not possess. Researchers found that many biological factors contribute to a bear's maintenance of their bones, such as their level of calcium. The bear's levels of calcium in the blood did not change during hibernation, meaning that even though bone remodeling is suppressed, it remains balanced, which helps limit bone loss. A look at one of the hormones known to reduce bone resorption showed that it surged to levels 15 times higher than normal in the bull zebras, which suggests that it may play a role in regulating the ways bears have evolved to protect their skeleton and bone resorption from the long periods of stillness. How does this benefit medical research? Many studies show that the possibility that elucidating the biological mechanisms that preserve bone tissue in black bears may lead to novel therapies for osteoporosis and medical research. Osteoporosis is common, subjecting approximately 10 million Americans to weak bones and placing another 44 million Americans with low bone density at risk of developing the disease. Finding treatment for this disease can save future generations from falling risk to chronic disease that carry an elevated risk of fractures, morbidity, and death. Conclusions. As this is a fairly recent new study, there is more to be enlightened about and more mechanisms to be uncovered and studied about. If there is continued research on these mechanisms, there could be a promising advancement in medicine and cures for osteoporosis. Due to this research, many other questions have arisen. Questioning if a black bear's mechanism during hibernation can be useful for other medical conditions as well. This includes the question of if researchers were able to find the potential genetic keys to a bear's insulin control. Could this be a novel therapy and treatment for diabetes? As these new questions arise and present, scientists will continue to conduct more experiments to find out about these mysteries about bear hibernation. Any questions?
So do we know, have scientists used, I know you said it's very new, but in terms of treatment for osteoporosis, do you see an application for, say, the hormone that you mentioned that was good at reabsorbing calcium or encourage calcium? Is there a way that we could maybe use that for treating osteoporosis? Is that the hope?
So right now, we only know very little about it, so we're trying to figure out more about how it works before trying to find a cure for osteoporosis.
So since not all black bears hibernate, do you know if this genetic marker that's activated in the hibernating bears, in your research, was it throughout the bear population, or?
I know it happens in grizzly bears too, but
Okay.
So today I'm going to be talking about the consequences of the Ohio train derailment. So you may be asking, what exactly happened during the Ohio train derailment? On February 3rd, 2023 at 8:55 p.m. Eastern Standard Time, 38 train cars derailed from the train line. In order to prevent catastrophic explosions, officials intentionally burned the cars. And while no explosions occurred, toxic chemicals were released as these train cars were carrying hazardous materials such as vinyl chloride and butyl acetate. So another question is, does it matter? Is it important? So long story short, it does matter. And I just want to show you guys a picture quick. There we go. So when vinyl chloride burns, it releases dioxins into the air. So this is an image of a man who was exposed to a high level of dioxins. Now these pictures were taken in the same year. This first one was taken in June, late June, and this is the 1st of November. You can see the effects of the dioxin on this man's face. You can see his whole color change. He has now scales on his body. And here's just another sign of a warning for these dioxins. So vinyl chloride and butyl acetate are both very toxic to humans. Vinyl chloride is a carcinogen, which is banned in all household products, as it can cause liver, brain, blood, and breast cancers. Butyl acetate can cause contact irritation, headaches, and respiratory failure. So now about these dioxins that are released into the environment. They can linger for 7 to 10 years before they finally begin to break down. Dioxins often contaminate crops, waterways, and they are also stored in animal fat while building up. So think about it, you eat animals, you eat vegetables, that is all ways that dioxins can enter our body. So the question's answered. So it's kind of hard to talk about this in full detail because this just happened last month, but there are some questions that have been answered by scientists who are conducting tests. So we know now how dangerous dioxins can be for humans. That has been known for a long time. Independent lab testing was done in March, earlier this month, and elevated levels of concerning chemicals were found. An additional chemical called acrolein was found in samples in East Palestine, Ohio, which is where the derailment occurred. It had the highest level present out of all the chemicals found in the area, even more than the vinyl chloride and the butyl acetate. Acrolein enters the body by inhalation, ingestion, or absorption through the skin and the eyes. Now, there's different reactions for different amounts that you are exposed to. If you're exposed to low amounts, it can cause your headaches, dizziness, nausea, fainting, things like that. But in high amounts, it can even cause death by respiratory distress. So obviously there are very many unknowns about this topic since, like I said, it happened last month. A lot of people are wondering, what's going to happen now? Where is it going to go? So while some officials claim that there is no threat to life, health, or the environment, many individuals in the area have very many symptoms, such as bronchitis, headaches, dizziness, nausea, and this has been lasting ever since the derailment occurred. There are no lab tests currently for humans in order to check if the symptoms are caused by the train derailment. We just don't know yet. It'll take some time, obviously, to evaluate all of the damage caused by the disaster. The effect of acrolein on humans in the area is unknown at this time. I'm sure things will come out hopefully soon. So obviously there are some questions that very many people have. Are these chemicals at a harmful level, or are the symptoms just coincidental? Are truthful details being covered up? Is the government trying to cover up exactly how bad this derailment was? Will residents get cancer from these chemicals in the future? Remember the vinyl chloride, a very, very dangerous carcinogen. How effective will crops and livestock really be? Again, it's going to take some time. The dioxins can build up in bodies for 7 to 10 years. So that is a very long time for these animals to be building up these chemicals in the fat in their bodies. And how far will the chemicals reach? This is a big one that a lot of people often talk about. It really all depends on the wind, but again, we don't know yet. It only happened last month. And a lot of tests are just finally starting to be conducted in the area. So imagine how long it will take for the tests to be out of the area. So hopefully these answers can be answered soon enough, because a lot of people are wondering about a lot of this. So I do have some pictures I want to show you after my work cited, so I'm just going to play those now. So there you can see the amount of smoke that was in the area. There's some of the scientists that are trying to test for these chemicals present. That's the actual derailment site where it occurred. And there's another image of the actual explosion. It did explode a little bit, but the actual fire where they were trying to burn off the chemicals. So does anyone have any questions for me? Yes, Ren?
What are the chemicals you name even used for?
So vinyl chloride is often found in things such as PVC pipes, which is actually, it stands for polyvinyl chloride pipes. They can be found in like things like wire coatings and that sort. Butyl acetate can be found in low amounts in some things we use daily, such as fragrances, nail polish remover, paint, resin. And acrolein is actually used to control plant, algae, and bacteria growth. It is also used to make acetic acid. Any other questions? Yes, Francisco Maker.
So, are they worried? It's hard to say because, like you said, it's so new.
Yes.
So we're worried about the aggregation of the chemicals within anything that's living in the area. Is there a different effect on, say, simpler animals, non-vertebrates, things that might be in the soil that's contaminated versus something that is living there but maybe it has like air contact with it, or something that's aquatic? Like,
Right, so you're
depending on the
asking,
environment.
basically based on the habitat, is there more of a chance for animals to, okay, that's a good question actually, because I did read up on this. So many of these dioxins, which is a very big concern right now, actually don't really spread throughout the air. So they mostly spread, like I said, through the crops, and they build up in the animal fat. So a lot of animals that have more fat, think of pigs or cows, things like that that humans ingest. It's found in higher amounts, along with aquatic amounts actually. There were tests done for some fish that were found dead near the derailment site, and they actually did have high amounts of dioxins in their body. So these are all things that, again, we ingest, and they are found in very high amounts, just not really in the air, because dioxins have a really hard time spreading throughout the air. Any other questions? Yes?
So are they doing anything to look at, should something like this occur in the future, what other options do we have to control a potential explosion like this?
So that's the thing is, so you're asking basically, are they trying to find a way to prevent this from happening again? So what's funny is they thought this was the best method to prevent something catastrophic from happening. The whole reason they burned these train cars is because they wanted to prevent explosions. But they didn't really think of what will happen with this combustion of these chemicals. They didn't really go into that. They thought this was the best method, and obviously it is shown that it is not. So I think, I'm not sure if they are looking at new methods now. I'm sure they probably are. But like I said, this just happened a month ago, and this was their best method. This was their best way of preventing something catastrophic from happening. Just sadly, it still happened. Yes.
Do we know who made that decision?
We do know that it was the government officials that were in that localized area. It was people with the environment, just basically people in that East Palestine area. They decided, let's burn the cars, let's try to prevent catastrophic explosions, because don't get me wrong, the explosions would be a lot worse than what it is now. But they were kind of just trying to figure out the best way to prevent something bad like that from happening. It's just sadly didn't work out the way they wanted to. Yes?
Do we know how the train developed in the first place?
I actually did not look at that. It could be anything, really. Train derailments occur pretty often, but yeah, I'm not sure. But sadly, it just happened to be a train that was carrying very hazardous materials. Yes, Max?
Do you know like how far like the effects are being spread like throughout the country and like the world?
So they do not know that yet because they're still testing in East Palestine, which is where it happened. And a lot of government officials are saying that there's no threat to anywhere. So the chances are of tests being done in an area like this it's going to take a long time. And sadly, some effects may happen before they do get tested on. It all depends on wind shifts and things like that it'll, time will tell. Any other questions? Yes, Mr. Barr.
How do you clean it up? Are they what are they doing to try to mitigate the disastrous effects?
So the thing is, you can't really clean it up. That's the bad thing is as soon as this combustion happened, this fire happened, these chemicals were released into the air, into the environment, into the atmosphere. And that's the bad part about it is there's no controlling it now. There's no way of, this fire was burning for days. It wasn't just that and it was over. Like this fire was burning for a very long time. And the more the fire burned, the more combustion happened, the more chemicals were released into the air. So that's a good question, but there's really no way to change it. Yes?
I just wanted to add to you, I did hear that they're removing soil, they're pulling up all the rails and they're pulling out the soil from underneath the rails, but it could be months of excavation.
Pulling up soil into the riverbed.
Yeah, it's really hard because it's everywhere now. It's everywhere. Are there any other questions for me? No?
All right, we're going to break.
Okay, so my presentation
conditions about removing carbon from the ocean. The transition's slow. Carbon in the ocean. As we know, fossil fuel emissions have played a monumental role on our Earth. Up to 37.12 billion metric tons of carbon is emitted annually. These carbon emissions have led to drastic environmental and climate changes. The ocean is an example of the severe effects taking place. About 30% of the carbon that's emitted atmospherically is absorbed by the ocean. The ocean's characteristics for high carbon absorbency led to many innovative ideas to be proposed. These ideas would overall help the ongoing issue of increasing carbon in the ocean. Effects on marine wildlife. Due to the high increase of carbon in the ocean, a detrimental effect has been occurring. Acidification. Acidification is the act of the ocean's pH lowering as the carbon intake rises. Acidification plays a substantial role for marine wildlife. One danger of this is it causing a decrease in calcium carbonate. This essential marine life building block helps aid aquatic animals in shell building and skeletal forming. Another ability altered by acidification is coral reef rebuilding. Coral systems are already extremely damaged, and a decrease in one of their main rebuilding components would overall inhibit their ecosystems more. Overall, oceanic carbon absorption would deeply affect marine food chains. Okay. So in this picture right here, you can see that the shells, this is due to acidification, and as time goes on, the acid basically wears down their shells because it's acidic, and then there's no calcium carbonate for them to rebuild their shells. So over time, shelled animals like crab, lobster, stuff like that, they're beginning to decrease because they can't make their shells like thick enough for them to survive substantially. Removing carbon from the ocean. Due to many problems arising from carbon emissions, scientists have been researching new, innovative ideas to aid in carbon removal. Ideas like electrochemical ocean capture and marine ecosystem restoration are just some of the few methods being proposed to combat this challenge. These methods, while being fairly new, could help revise and prevent further damage to marine biology. Electrochemical ocean capture. Electrochemical ocean capture essentially uses electrical currents to alter salt and water molecules in ocean water. This helps create a basic and an acidic solution. So basically, an acidic solution will wear down alkaline rocks in the ocean, like on the floor, and the alkaline rocks will increase the alkalinity. And the basic solution would also help increase the ocean's alkalinity by absorbing carbon, and basically it stabilizes it and neutralizes it. So this stabilized carbon then turns into bicarbonate ions. This method would effectively increase the ocean's pH, which would counteract the active acidification. This would benefit marine life in the future. In order for marine life to live in the ocean properly, the ocean has to maintain a certain pH. Once that pH is thrown off, all of the marine ecosystems, they're thrown off too, the animals are affected. So this method would essentially just increase alkalinity, which would help maintain the pH levels. Next one. Marine ecosystem restoration. Many coastal marine vegetations absorb and use carbon in photosynthesis processes. Most of these vegetations hold a significant amount of blue carbon, which refers to large amounts of carbon stored in these ecosystems. When these ecosystems are destroyed, that carbon is no longer stored but instead released. So it's just basically like, if there's no coastal vegetations, because they're constantly being destroyed, all that stored carbon, which the vegetations are holding, are then released into the atmosphere, and then that's absorbed by the ocean. So one step they wanted to do was start the protection and restorations of marine ecosystems, which would help prevent carbon from being absorbed atmospherically and aquatically. This method is seen as a reliable and efficient method due to environmental co-benefits, while also efficiently preventing oceanic carbon absorption. Here you see there are lots of teams rebuilding marine ecosystems, so that's the top picture. And there are teams going around which are rebuilding destroyed coastal environments. How this helps us. These new methods and research are helping lead to a bigger picture, a cleaner Earth. This revolutionary research will allow scientists to tackle carbon pollution on all fields instead of just atmospherically and terrestrially. Overall, slowing down climate change and making a safer environment for us and other life. This study will help protect the marine life we so heavily depend on and soil acidification. This process would essentially make it a cleaner environment for the marine life to live off of, and it would also help us because we rely on these animals, so if there's a decrease in them, that would also affect us, and it would affect human food chains and stuff like that. Here's my works cited. Does anyone have questions?
Where'd you come up with this topic?
One question was, where'd you come up with this topic? I searched it up, and it was fairly new. And they haven't really done any of these things yet, but it's being proposed, and they're trying to test things out.
Thank you.
Any other questions?
Did you read about anything specific to New York? I mean, we have a lot of ocean coastline, we have the Hudson River, which is an estuary, or like the DEC or the...
Okay, did I read about anything specific to New York? I know that they have just started proposing this actually being used. So this is mostly a fairly new thing. It hasn't really been taken out of the lab yet. But I think that they're going to start working in areas that are seen to be more, because there are lots of areas where shellfish live, and they see that those areas are being affected the most because there's a decrease in these animals. So I think they're going to start working there to kind of undo any damage that was done, and then they're going to move on to other parts of the ocean that have severe carbon pollution. Any other questions?
Are there any recommendations you could make? I mean, if there's severe carbon pollution, are there things that we could do that would limit the amount of carbon that's being put into the atmosphere?
Well, that's just there's small great things that we can do that could help, but most of the carbon that's being emitted is done by large, something that I saw was that people can help a little bit in reducing carbon emissions, but it's the bigger things like jet fuels and cars. Everyone drives a car and they all emit carbon dioxide. That plays a huge factor. Factories. So there's nothing really that, it would have to be a bigger step. Like an individual person can't help anything.
Can we use biofuel?
Yeah, biofuel would help. Maybe my presentation should have been after his presentation.
So my presentation was on exploring the mineral effects of cannabis. The history of cannabis. The initial cultivation of cannabis began in the 1600s. It was used to make rope, sails, and clothing due to its unusually strong fiber. The government actually encouraged the production of cannabis for this purpose. Medical use of cannabis became popularized between 1850 and 1937. It was widely used as a painkiller that could be easily purchased in general stores and pharmacies. The Marijuana Tax Act of 1937. Research linking cannabis with violence, crime, and other social issues in the 1930s instilled a fear within the American public. National propaganda against the evil weed ultimately encouraged the government to take action against cannabis, resulting in the Marijuana Tax Act of 1937. This act effectively made the drug illegal across the country. Legalization of cannabis in New York. Medical cannabis became legally available through the medical marijuana program in 2014. New York then legalized recreational use in 2021. The behavioral effects of the psychedelic drugs. What are psychedelic drugs and what do they do? According to the American Addiction Center, psychedelic drugs are a subclass of a broader class of drugs that are called hallucinogenic drugs. These drugs alter one's cognition in a way that is different from how other drugs alter it. For example, central nervous system depressants and stimulants only amplify already familiar states of conscious experience. However, psychedelic drugs alter an individual's consciousness in a way that they are no longer in a familiar state, but are in new states of consciousness. Is cannabis a psychedelic drug? There is a common misconception that cannabis is a psychedelic drug or hallucinogen. Cannabis actually belongs to the cannabinoids drug class, which does exhibit some hallucinogenic properties, but it also exhibits some properties of central nervous system stimulants and depressants. Professor Natasha Mason's research on cannabis. Who is Natasha Mason? Mason is an assistant professor in the Department of Psychopharmacology at Maastricht University in the Netherlands. Why did she conduct this research? Mason wanted to explore the acute effects of cannabis on the brain as well as the long-term effects of cannabis on brain on behavior. How did Mason conduct this research and what did she discover? Using magnetic resonance imaging, or MRI technology, Mason examined the brains of individuals who use cannabis on a monthly, weekly, and daily basis. For the acute aspect of the research, Mason would vaporize cannabis and have the patient inhale it while under the MRI machine, specifically observing how certain changes in the brain relate to the feelings of subjective high and behavioral impairment. She also took blood samples from each patient. In doing so, she discovered the drug needed to pass a certain threshold within the blood in order for the person to experience the high or show an impairment or the behavior differences associated with the drug. Mason also performed the same test on individuals who smoke cannabis daily. These individuals had actually developed a tolerance, so they were not experiencing the subjective high or behavioral impairment. Why does this matter? Medically, cannabis has become a widely used drug. Many people use cannabis to control pain and relieve anxiety. These people likely don't want to experience the associated high or impairment. With this new information, we may be able to determine a certain dosage that will be useful medically while not inducing the unwanted high or causing impairment. This research is also significant for recreational users. Since we now understand that a certain tolerance to cannabis may develop, recreational users might consider limiting their exposure. Cannabis becomes especially detrimental when people become addicted to it, as the CDC states that addiction results in problems with attention, memory, and learning. If recreational users continually use the same amount, they will likely develop a tolerance over time, meaning they will need more and more cannabis to induce the desired high. To prevent the risk of developing an addiction, recreational users might be able to take break days in order to avoid becoming tolerant and thus reduce their likelihood of becoming addicted. This would result in a safer, more informed way of using cannabis so it is not detrimental to their well-being. Additional questions the research introduces. What dose of cannabis and what use frequency is required to produce tolerance? Now that we're aware that there is a possibility to produce tolerance, it raises the question as to how much and how often it needs to be used to develop this tolerance. This again is significant for both medical and recreational users, as it will certainly help guide how much cannabis should be used for each desired effect. What effect does a built tolerance have on a person? Due to the uncertain nature of how this tolerance forms and how cannabis affects the body systems in the long term, we are prompted to consider what effect this tolerance may have on our brain and body. My thoughts about the research. These findings are very significant and intriguing discoveries that may guide future studies to help us truly understand the effects of these drugs. The research provides valuable information to recreational users and may even aid in the prevention of addiction. It also provides a promising outlook for medical cannabis, as it may be possible to isolate its therapeutic effects and negate its impaired ones. While there are certainly many more studies to be conducted to fully understand how cannabis affects the brain and individual, this research certainly serves as a significant checkpoint in our ongoing analysis of cannabis. And these are my sources. Questions? Mr. Duchamp?
Can a tolerance be built up for the pain-killing effects in patients?
So the research didn't specifically state that there was a built tolerance for the medical aspect of using cannabis, but it did state that over time, people who use cannabis continually develop a tolerance, and we're not completely sure why they develop a tolerance, but it is just known that they do develop a tolerance. And part of the reason why they can tell is because when they were examining the brain in the MRI machine, they discovered that a certain hormone that was usually released when the cannabis had its effect on the brain was not released in people who used it long term. Yes?
Did they actually consider it an addiction, or is it more of a dependency? Or was there like any research that classified it as an addiction?
So the question was there any research that classified cannabis as an addiction or a dependency? I don't believe my research included any information on that, but the Centers of American Drugs or American Addiction Centers stated that cannabis could induce an addiction. So based on that, I would say it is possible that there is an addiction. Yes?
So you said there's detrimental effects of cannabis. Is there a difference with recreational use versus medical use, or is it both the same, like the effects, the detrimental effects? Is it the same for both?
Okay, so the question was, are the effects of cannabis the same for medical use and recreational use? And they are the same. So as I was saying earlier, a lot of people use cannabis for pain and anxiety, but along with that relief of the anxiety and pain, there's also like the effects of impairment that recreational users also face. Yes?
Do we know exactly what chemical is released?
The question was, do I know what chemicals release? And I believe it was some kind of hormone that was like a pleasure-inducing hormone. Yes?
You mentioned some of the negative effects on long-term users. Can you elaborate further on that?
So I stated that the negative long-term effects on users, as you asked, the American Addiction Centers stated that I have it in here. So the CDC actually stated that addiction results in problems with attention, memory, and learning. My research wasn't exactly on like the detrimental effects of cannabis, so I was not able to research that aspect. Any other questions? Shakib?
Going off of Heather's question, based on your research, do you think the ZA's worth it?
Personally, I would say that's something for you to consider, weighing the benefits and cons, it's up to you. Yes?
Do you think that recreational ZA should be legal?
So, I mean, to each their own, right?
No spoilers. No spoilers for The Last of Us, please.
Zombie plague.
All right, zombie plagues. Famous fiction or fitting fact? I am Ren, by the way. I was just told that I need to start introducing myself, so. So this presentation is based off of The Last of Us, if you could not already guess. If you have not seen The Last of Us, you should. But in the show, humans have been affected by the very real Orpheocordyceps unilaterus fungus. Please don't make me say that again. From now on, it will be known as the zombie ant fungus. The show points to global warming as the catalyst for this fungus to be able to infect humans, when otherwise it would not be able to withstand the high body temperatures in humans. So how does this fungus work? Typically found in rainforest climates, the ant must first come into contact with spores from this fungus, whether through foraging or from another infected insect, where the spore then burrows through the exoskeleton, breaking down the chitin there, to then become in the host and start growing there. In the beginning of infection, the ant reacts normally. This is so that the ant never actually gets kicked out of the colony like a normal sick ant would. However, after time, the ant becomes hyperactive and it wanders away from the colony, where it goes around 10 inches above the forest floor and finds either a stalk or a leaf and bites down on the middle vein of it in what is called a death grip. This is called a death grip because even after the ant dies, it will remain just as strong as before. Terrifyingly, it has been just recently found out that this fungus never actually infects the brain. In fact, it hijacks motor control, leaving all mental capacities just the same. So after the ant dies, the fruiting body erupts from the top of the head and releases the reproductive spores onto the colony below. You can see kind of the life cycle on the right in a cartoonish image, and then on the left, real-life examples where you can see the fruiting body, the death grip, and just the dead ant. So how do fungus affect humans right now? Fungal infections can be anything from the kind of gross but minor athlete's foot to terrifyingly deadly yeast infections where 1 out of 3 patients will actually die from them. They are majority airborne diseases spreading through reproductive spores that come into contact with either the skin or the airways, and it depends on the species. Candida auroris. Candida auroris is a new species of fungus that popped up right before COVID. And in fact, over here you can see a map of 2022 of people infected with this disease, with the dark orange red being 300 cases or more. And this is one of those terrifyingly deadly diseases where 1 out of 3 patients will die when they contract it. It spreads through skin contact, and it is extremely resistant to antifungal drugs. However, what is most important about this is scientists believe that this is the first of what could be many of fungal infections that have actually evolved to withstand higher temperatures because of climate change. And they believe this because this fungus started affecting people all at once across the globe. This did not start somewhere and then spread everywhere else like COVID-19. This just started infecting people all at once, like it just recently evolved to do so. So we kind of have the perfect storm. So are zombies possible? The short answer is probably not. And certainly not something that this generation has to worry about. Because the zombie ant fungus is a zombie ant fungus. It has spent countless years of coevolution with ants, specifically formicide ants, which is a subfamily of ants, with very few discoveries outside of this subfamily and very rare cases of other insects. This fungus has never been seen to infect anything outside of the phylum Arthropoda. So no amphibians, no reptiles, and certainly no mammals. So it would be hard to believe that anytime soon that this fungus would begin to start going after our highly complicated immune systems. These are my citations, and any questions? Yes?
So, with no spoilers, but just a character in the show Shot's game believes that the people are still alive inside when they're affected by the fungus. Do you believe, since it only affects the muscles, if someone got infected, do you believe that they are conscious on the inside as they're being infected by a fungus?
That could be possible because the fungus never actually attacks the brain. However, it would be probably not something you would survive for long, as you wouldn't be getting the needs that the human body would require to continue living. Yes?
So without controlling the brain, how does it take over the ants, like the muscle system?
From what I read, it can be kind of assumed that it kind of hijacks either the nervous system or the actual just basic motor functions of the ants. However, that was not something that I specifically went to research on, as it was just something that kind of popped out as I was researching very new information. So that would be something that I would have to look more into. Any other questions?